so we don't need, so we all love bats, so we don't need arguments to help for that conservation, to make surveys, to develop measures for that conservation. But unfortunately, governments, stakeholders, they need a bit of motivations, and one of the motivation is money. So, and we talk about money and insectivorous bats. So we think about ecosystem services. So I will start to talk about a little, just to introduce ecosystem services, and after I will go through the our Irish bats. So I can, it's okay, Karen. You can see the slide just to double check.
Yeah, I
you see it
see it.
perfectly.
Okay,
Thanks.
perfect. So just yeah, let's talk about insectivorous bats in general. So, and why we at the moment there's a lot of studies on ecosystem services. So there is close 1,500 species of bats worldwide. So it's a lot, and 2/3 of them are insectivorous bat species. So you can see on the slide there some species. So here, so this one is from France, this one from Ireland, and here this one, for example, on the left, on the right, it's from Africa. I have some from Panama. So a lot of insectivorous bats, and they all have a very broad diet. So they consume flies, bugs, beetle, moth, spiders, a lot of species. And they also use a lot of landscapes. So wetland, pasture, crops, plantation, forest, lake, urban area, and even desert. So, and because they use so many habitats, they have a very diverse diet. So potentially they can eat forage on pest species. And this is where ecosystem services pop in. So what is ecosystem services? So there are the benefits that people obtain from ecosystem services. Ecosystem. So it could be a species, could be ecosystem in general, could be a taxa. And so because they hit so many species, so many prey, so there are a lot of studies from the last to 10 years, 10 years, 20 years. So lot of studies, and they confirm that they eat a lot of prey species. So some species are impact livestock, for example, so cows or sheep, goats, but also crops. And when we talk about ecosystem services, if we find arguments, but we can find arguments for bat conservation, like I said before. So there are also studies that develop the idea that if we conserve the bats, we will help to conserve the ecosystem services, so it will be useful for human society. So what do we know about ecosystem services now? So this map shows a number of studies worldwide, so depending on the country. So on the the green, sorry. So light green means that there are few studies per country, and very dark green means that there are a lot of studies. So we can see that in Europe, so there are few studies, and it's not easy on this slide, but we can see Ireland, it's a very light green. So there one study on ecosystem services. But majority of the studies were done in the United States. And another thing interesting, it's the pie chart. So the pie chart indicates the dominant landscape in the study. So if, for example, if the study is focused on plantation or pastureland, urban area. And if we look at the pie chart, so what we can see is majority of the studies are done in arable land, so crops. And so it's good, I guess, but though that it's relevant for Ireland. So in Ireland, so I guess you all know that, majority of our landscape is pasture. So 59% of our land cover is pasture. So it's very, very high if you compare to Europe. And after the second is wetland, and again, very, very high percentage if you compare to Europe. And but just remember what I said before using the map. So there is very few studies, only one in Europe that investigates diet on pastoral habitat. So at the moment, we don't have a lot of knowledge on the ecosystem services that insectivorous bat can provide in pastureland. So yes, so we need to answer that. So for Ireland, but first, to answer these questions, what do we need to know? We need to know the diet of our bat species. So what do we know? So just talking about Irish bats, so 9 resident bat species, the soprano, the pipistrelle, some myotis species, one horseshoe bat, the lesser horseshoe bat, and the brown long-eared bat, and the lesser bat. And of course, two other migrant species. So our bat species, they use different kind of habitats. So of course pasture, because it's full of pasture, and but with cows and also sheep, some forest, other wetland area, even mountains, and also of course here on the left top, it's a Phoenix Park, so it's in urban area. So all these bat species, they use different, bit differently as habitat. So some of them, like the lesser bats, they will forage in open area. So between trees areas, or on above trees, or even completely open area, rivers, lake. They love that. Other, for example, and other species like the soprano pipistrelle, they will forage on the edge of the vegetation, and other bat species like the brown long-eared and the lesser bat will they will prefer forage very close, even into the vegetation, in narrow space. So depending on the foraging behavior, they can eat different kind of prey, and of course pests. So this is kind of important for after. So what do we know about the diet? So there are a few old study. I'm sorry for the term old study, but it's they were done, I think, for the 2021 for the Dobunton. So, and these four studies were based on morphological analysis. So we learned a lot from them because it was the first studies in Ireland looking at the diet. So we have knowledge on the Leicester diet, the Natter, the Bourgogne bat, the Daubenton, and the Leicester bat. But so we know that they eat a lot of prey, beetles, mosquitoes, flies, crane flies, spiders, moth, beetles, even Hymenoptera. So, sorry, what the name? It's not ants, but Hymenoptera. Sorry, I don't remember the name in English. Sorry, wasps. Sorry, sorry about that. And but these studies use old technique. So morphological analysis. So you have to imagine. So all these people, they collected a fecal sample, and after they look into each fecal samples and look for remains of prey. So they have to look in microscope and try to identify species from, for example, legs in the A, so top left, or from a wing. So it's a lot of work. And sometimes, so I guess at the end, they were all a bit like this, very tired. And one of the issue with this technique, so it's we can't really, so for majority of taxa, we can't really identify the species level. So it was more, the prey were more identified at genus or family level. And now we have a new technique, and this technique was used in only one study in Ireland on Leicester bats. So they use metabarcoding. So they use DNA. I will just explain after that, just how it works. So, and this technique allows to identify the prey using DNA found in the fecal sample. So, briefly, so the bat eat the prey. The prey goes through the digestive system and ends in the poo, and we use the DNA from the poo to identify the species. So just a quick overview of how it works. And please, at the end, if you don't understand, if it's not clear enough, I will be very happy to explain again or provide more details. So how from the poo, we can identify the species. So it's important to say that in fecal samples, so the DNA is very degraded. So it went through the digestive system, so with a lot of enzymes. There's even a lot of bacteria. So it's not very good DNA. It's very dirty. So it's one of the issue with this kind of technique. So usually when you want to identify a species, we use nuclear DNA. But nuclear DNA, it's I don't know if you can see, laser here. So nuclear DNA, there's only one copy by cell. And because the DNA is very degraded, if you're not in lock and the DNA it's degraded in the nucleus, you won't be able to identify the species. But in the cell, there is other organelle, so mitochondria. So mitochondria is these little tiny things, and in each cells, you, me, everybody, every species, we have a lot of copies of mitochondria. And mitochondria, they have a DNA. So they all have the same DNA. So, and because we have so many copies of mitochondria DNA, we can use this DNA because a lot of copies. So it's the thing with the DNA we use in this kind of method. So we use a small portion of the DNA and we use like a barcode, okay? And so we extract the DNA and after we amplify the DNA in all fecal samples. So we have all this barcode. So of course, after the lab work, we don't know what is the species, but after we send the sample in sequencing company and after we run a bioinformatic pipeline, and at the end of the bioinformatic pipeline, we are able to identify, so this barcode is the bat, for example, and the blue barcode, it's a mosquitoes. The orange, a brown barcode, it's, I will say, tipula, so crane flies. And the green, it's a moth, and the dark green, it's a spiders. So it's just how the metabarcoding works. So it's very useful to identify even a tiny level of DNA from the fecal sample. So it's very, very powerful tool. So just to come back to my first question, what are the diet of bat species in Ireland? So, so we have very old studies, so based on morphological analysis, and we have only one study based on new technique and very powerful technique. So what do we know? We don't know a lot, to be honest, at the moment. So it's why my project came up. So my main project was to characterize the diet of two common Irish bat species, so the brown long-eared bats and the soprano pipistrelle. So why these two species? Because Karen told me that a lot of you present tonight, so you're doing brown long-eared bat surveys, so you know that these bats they will stay in the roost, and in most of the roost, so we can go in the roost, and but these roosts are very stable from May to August. So it's it's easier, I would say very easy, I would say more easier to collect a fecal sample from this roost. And also, why the soprano pipistrelle? So soprano pipistrelle is a bit more tricky to collect fecal samples, but we choose this species because it was nice to have two different foraging behavior. Talk about this before. So two different species, two different foraging behavior. So potentially they have a different diet and they provide different ecosystem services. So so we start the project so we start sampling in 2021. So we selected 19 bat maternity roosts all across Ireland. Pretty sure you can know some of them on the screen. And we collected the feces for 3 years, so from 21 to 23. And 3 time points each year. So during the gestation, so mid-May, mid-June, and lactation July, and post-lactation in August. So why we did that? Because we wanted to see if, depending on the period, the diet differs. And it was very important to use non-invasive sampling. So we use non-invasive sampling. I will show you just after using pictures how we did that. So so on the map you can see all the location of the roost. So yes, so you can see some soprano pipistrelle and brown long-eared bat. So sometimes it's very easy. You can see them very easily, and and sometimes it's a bit more tricky, especially for the soprano pipistrelle. And how we did that. So we use newspaper, very easy. Everyone has newspaper. And we laid on the newspaper for 2 days. So laid on the newspaper and we came back 2 days after to collect the sample. So you can see some examples. So here you can see some fecal sample here. Sometimes it's a bit more tricky. For example, here on the top right, I had to use the pipes in the attic. Sometimes we have very few droppings, but sometimes I was very lucky with a lot of droppings. So it was not difficult for the Bolognier and sometimes okay for the soprano pipistrelle, but for one roost we struggle a little bit. So maybe some of you know this roost with Drummer Wood in County Clare. Lovely landscape in woodland and lake. But the soprano pipistrelle, they use the roof, but the exit is here, and there is no way for me to go into the attic. So I had to find a way, and I use an outside collection. So I set up a little platform like this just at the exit of the roost, and also I put a newspaper directly on the wall, and you can see here some fecal sample. Because the droppings are a bit wet, so it works very well. And it was good to know that we can have a lot of droppings even using outside collection. So in total, for 3 years, we collect more than 5,000 droppings. So it was a lot of tubes, but we were very happy. So we got all these droppings. After, unfortunately, we had to select the roost because it was important that we needed for each roost. We needed for each event we needed fecal samples of what happened for some roost. So for example, in 2021, we had a lot of droppings and nothing in 2022. And because we wanted to analyze the diet for the 3 years, there's unfortunately some roost we had to discard from the analysis. But we still have the dropping, so we can still come back to them. So at the end, I will show you today the analysis of 12 roosts, so 8 Bolognier bats and 4 soprano pipistrelle. You can see them here on the map. All around here, it's the landscape across around the roost. In green, so just like a confirmation that green represents grassland. So wet grassland or improved grassland, but grassland. So, and again, we can see a lot of grassland around each roost. So very specific to Ireland. We won't see that in France, for example, or Portugal or Germany. So what did we find? So we found a lot of prey species. So close 400 prey species for the brown long-eared bat and 364 for the soprano pipistrelle. So here are some examples. So you can have some beetles, moths, spiders, some mosquitoes, flies, micromouse for the soprano pipistrelle. So a lot of species, and we will go into more details here. So this graph represents all. So here you have all the orders identified. So we have some queen flies, we have some moth, isopoda, and some Hymenoptera and bugs, flies, beetles, spiders, and the soprano are represented shown on the left, and the brown bats on the right. So bigger is the circle, more there is species by order. So for example, there is a lot more than 200 species of flies for the soprano pipistrelle. A bit less for the brown bats. It's between 100 and 200. And the color represents the average of species for each order by droppings. So, for example, so in for the soprano pipistrelle, so it's the mouth in purple. So it means that between around two more species per fecal sample, and more for Diptera, more than 5. So you can see the two main orders, Diptera and flies and moth species. We also have some non-flying insects like spiders. It's very interesting, and even some for soprano pipistrelle, so that we're not surprised for the brown bat that if we have non-flying spiders in the diet, it was more a bit surprising for the soprano pipistrelle, but yeah, why not? So after I also wanted to identify how many species we have in the diet. So how many species by fecal samples? So do they eat a lot by night or not? So for the brown bats, so the range is very wide. So I found between 1 to 20 up to 21 species by fecal samples for the brown bats, and in average 6.3 species by feces. And it's a bit similar for the soprano pipistrelle. So 1 to 20 and 7 in average. So here can see so 1 dots, it's 1 fecal samples, and it's the richness for each fecal sample for the 2 bat species, so soprano on the left and the brown bat on the right, and for each host. So, and the colors indicate as the year of the collection. So can see that here, so there are few majority of the fecal samples are below a 10, and there is some with like carrot gems are a very high richness sometimes, but most of the roots are very low richness. And what is interesting, it's lower than in European studies. So why? It's a good question. We have some ideas, but what is interesting too is I talked about this paper before. So it's so in this paper the Karen Chill, in her team, they investigate the diet of the Leicester horseshoe in Ireland, but they compare the diet with England and Germany West. And what they found that in Ireland, the richness by fecal samples is lower. So we find than in England, in Germany. So we found the same pattern. So in Ireland, apparently, all bats eat less prey species. So why? Could be an impact of the Irish landscape. For this, we need to understand a bit more. But there's also another possibility that we have a lower arthropod diversity in Ireland. And we know that. So maybe it's linked to that. But it's very interesting that we found the same patterns between the two studies. So I was also interested to look at the diet composition. So this plot, when I talk about diet, so we talk about richness before, so how many prey per fecal samples, but what is also interesting is how the diet varies across the roost, across the species, and even across the year. So this is a very busy plot, but I will just explain. So it's just to show the similarity in diet between the two species. So one dot, it's one fecal sample, and if two dots are close together, it means that the diet composition is very similar. So it means that they eat roughly the same prey species. In contrast, if two plots are very far away, it means that the prey that we found in the diet are very different at the species level, very important. So first, what we found that so the soprano are in a light green and the Bois bat here in a very dark green. So the two bat species, even if they share, if they hit more deep flies and moth together, they don't hit the same species, so it's very interesting. Statistically, the two composition for the two species are different. So it was kind of, so of course we expected that a bit, but it's nice to see that on the results. We also look at the composition between the roost. So yeah, clear effect on that species. So I'm sorry, it's still very busy plot. I did my best, but so each color represents a roost. And what we can see, it's depending on the roost. So for example, Kinvara here, and we in pink and blue, so we have Litterfrack here. So these two roosts have a different composition. So there is very few overlap, few blue here, but very few. So these two roosts, they don't have the same composition. Some roosts are very clustered together, but other roosts have composition, diet composition more, it's more spread out. If I say so. So for example, Glengarry here, so you can see dots of Glengarry, so it's this green, they're a bit everywhere, here, here, here. But again, for example, Kinvara, they're all together. So again, depending on the roost, the composition varies a lot. But let's see what are the percentage of each order for the Bois bats by roost. So this is a web plot. So it shows the interaction, so meaning predation on the roost for each roost with the orders. So when I say predation, it means it's if I found one species in the diet, it's one interaction. In one roost, it's one interaction with one species, so between the roost and the prey species. Prey species together by order, we have this plot. And bigger are the bar here, more there interaction between the roost and the order. So for example, in Cara James, so the moth, there more interaction between the bats in this roost with the moth than with the Diptera. So again, we're not very surprised because we know that the Bois bat, it's well known to eat a lot of Lepidoptera moth. So it's the case for a lot of roosts. So for example, so the Lepidoptera are the main, the dominant order, but only with 61, I will say, a percentage of the diet. So most of them are Noctuidae, so like the lesser yellow underwing or other most families. However, the flies here in purple represent a lot in the diet. They represent 31%. So it's a lot for a species that's well known to forage on moth. Most of them are crane flies and flies and even dung flies. But I want to highlight two roosts. So Glengarriff. So I guess you know Glengarriff, and it's an amazing place. Very natural landscape vegetations. And in this roost, we can see that the diet is the most diverse. So some beetles, and even some bugs. So even if the moths are the dominant, Diptera also, but it's a bit more diverse than if you compare with the other roost. So it's interesting. So it means that when there's other orders present in a rear, the Bologna bat will also forage on them, not only on the moths and on the flies. And there is even more interesting, Kilmore. So Kilmore is in Cavan, and for this was there is a switch. So see the purple. So it's the interaction with flies are more important than the interaction between the moths. So it was very interesting. It means that the Bologna bat it has foraging flexibility. Again, at the moment with our data, we don't really know why. It's I would say, an open question for future. Why in this roost they completely switch? It's very interesting. Anyway, so here for the Bologna and for the soprano pipistrelle. So less roost, only 4. But again, so if we come back to the dissimilarity plots, so we can see that so there is 3 roosts clustered together. So here, Burr, Kilafin are clustered together, Drummerwood, but Glendalough, they are very more widespread as the fecal sample. So it's like there's some overlap with this roost with the other, the composition of the diet from the other roost. But still, statistically, this roost have a different composition. So again, it's very interesting. They are very adaptable depending on what are the species across the roost. I guess they forage what they can find close the roost. So if we come back to the same interaction with plants, so this is the same. So it's showing the predation between each roost and the order. And here a lot of purple. So we can clearly see that majority of the prey are flies. A lot of non-biting midges, Psacoda, but also other flies and some mosquitoes, and also biting midges. The moths are present, but much less than the flies. So the Diptera represent 83%, so close 80, even 84% of the interaction. between the bats and the prey, and the most is the second dominant order. Is the second order. It's only 10%. So they have a very high. They need diptera. They need flies. It's very important for them. For the soprano pipistrelle. Just what I found interesting for the pipistrelle. So they hit very tiny images like this, 1 millimeter. It's very, very tiny, but not sure we can really see on the slide. But here's a picture of Tipula maxima. Tipula maxima, it's a big crown flies, 3 millimeters, 3 centimeters. So it's a huge prey. It's, we'll say, the size of the body of the pipistrelle. So they're not afraid to catch a very big prey for their size. So it's very interesting for this species. But again, a very high reliance on flies species for the soprano pipistrelle. So talk about the composition for each roost and talk about the richness and the main order. I told you that depending on the roost location, there is a different composition. So we wanted also to see if depending on the year, there is a different composition, if the year has an effect. And so what we did, so we did this kind of clustering method. So I will explain. It's not a difficult, could look a bit tricky, but it's not. So this it's a clustering. So it will cluster the composition with together if the composition is similar. So, but I just try to cluster so by roost and by year. And so what we found that what I will expect that depending on that the lawn cover, so the roost has bigger impact than the year. So I will expect that, for example, where is it? So I have a group, for example, Gwenaëlle, it's Burr, for example, here. It's a good example. Burr all together. You see? So I will expect more this kind of pattern. And what we found that the roost has an impact. For example, Kinvara here in 21 and in 22, the diet compositions are similar. However, for 23, so where is 23 for Kinvara? I lost Kinvara, but it's not here. No, where is it? Here. So it's very far away. So the year also has a very important impact, and and it's the case for most of the roost. So at the moment, unfortunately, we didn't have enough power in our statistical analysis to know if it's more important that the year has an impact or the roost has an impact. But it's still very interesting to see that. So, what has the diet of bat species, which was the first part of the project? So we learned that the Gwenaëlle bat has a foraging flexibility. Soprano pipistrelle shows a very strong preference for flies. And the richness, the composition of the diet is influenced by the lawn cover around the roost, but also the year. Yeah, and we think that it's because so it depends on the prey availability. So so an opinion, it's the brown longyearbot and soprano, they will find so why it's so variable across a year. And roost, they will just eat what they found. Of course, they have some preference, so flies for soprano and bit of mix for the brown longyearbots, but they will find what they have around the roost. Do they consume any pest species? So it was another very important part of the project to find an argument for their conservation. So, and the good news is yes, they eat a lot of pest species. So 30 pest species, and 6 of them are major pests in Ireland, so recognized by the government. So this two plots shows the number of species, pest species, by category. So we have crop and pasture, crop and vegetables. Majority of them are moth. It's why for the brown longyearbot here we have more species than the first soprano pipistrelle. Also some forestry species. Again, they're also moth species, but also human health. So in human health, there are mosquito species. So it's interesting. We don't have a lot of mosquitoes in Ireland. I guess it's why we don't have a lot of human health species in this plot. Also, livestock pest. There are more midges. I will talk just about this just after. And plantation. So plantation, it's between category, it represents apple tree, for example. And again, a lot of them, more of the species are moth species, so it's why we have more pest species in the diet of the brown longyearbot. Have a look per species. So for the brown longyearbot, so majority of the species, pest species are moth here, big majority. And we have this interesting guy. So it's Epiphias presbytana. So it's pest of apple tree. So recognized in Ireland, there is a survey. It's important. We also have 2 tipula species. So they are daddy long legs. 2 species. Again, I will talk about them after. And also some beetle species. For the soprano pipistrelle, so look, we also find again our 2 daddy long legs, the Tipula paludosa and Tipula oleacea. We also find the same moth species that feed on apple trees, on the leaves, but we also find some interesting species, culicoid. So culicoids are biting midges, and they are responsible of some important disease, for example, the blue tongue disease. So it's a horrible disease with a very high rate of mortality for the sheep. But it's very interesting that pipistrelle only, not the brown longyearbot, feed on this species. So see, depending, so 2 bad species, 2 different pests, different pest species. Of course, some are common, but also different. And it's very interesting because it means we have to protect the 2 bad species to provide ecosystem services, not only 1 species. So now let's come back to these 2 guys. So Tipula paludosa and Tipula oleacea, so daddy long legs. And they are major pests in Ireland. So the larvae, like you can see on the right, they feed of grassland. And so a lot of grassland in Ireland because we have a lot of dairy production, beef meat production. And I don't know if you know the economical importance of this, but in 2022, it was close to billions for the dairy production. So it is a lot. And for beef, it was around 3 billions. So if we don't have healthy pasture, we will lost productivity. And also another important thing, so until 2019, so there was one pesticide used for these two species, but it was horrible. So now it's banned in a lot of countries and in Europe. So we don't have any method to decrease the population of these two crown flies. But we found them in the diet of bat species. But what is the frequency? So FO represents frequency of occurrence. So it means that in how many fecal samples we found this one or two bat species. So in the brown long-eared bats, we found them in 49% of the feces and 19% for the soprano pipistrelle. But what is interesting is for for both species, we found them in 100% of the fecal sample. So somewhat, and for some event, sampling time. So it's very interesting. So let's have a look a bit closer to this. So this plot represents as frequency of occurrence. So one dot is one event. So for example, it's in gestation in 2021 for Gwenaëlle, for example. And so we have up to 9 dots per roost. So we have the soprano here on left and the brown bat on the right. And I organized the roost by increasing percentage of grassland around the roost. So for example, bear here, it's more on the right because a lot of grassland, 73, and Gwenaëlle, very, very few grassland in Gwenaëlle. So the same for the brown long-eared bat. And what we can see, it's when the percentage of grassland increase, the frequency of occurrence in average increase too. So it means when there is pastureland, so potentially more crown flies, the bat species, they will feed on the crown flies. So it's very interesting. So you can see here, so for some event, we have 100% of occurrence for these two species. So they eat a lot of crown flies. So it's very interesting. It's a very good point for the bats in Ireland. We were very, very happy. So just want to say that I was very lucky because I also very it wasn't the plan, but in some fecal sample were a lesser horseshoe bat sample. You know, sometimes in the roost you have some individuals, they are not the same species, and they're within the same building. And so I got lesser horseshoe bats and Natter bat. And I also find a lot of two pet species in the lesser horseshoe bat and the Natter fecal sample. So, and even for the lesser horseshoe bat, in 79% of the fecal sample, I found one of these two species, and 43 for the Natter bat. So it's a lot. It's very, very good point for the Irish bats. So just take home message. If someone says that the brown long-eared bats forage mostly on most species, Yes, but you can discuss, you can say yes, but in Ireland, in some place, it's not really true. They also forage a lot on the fly species. Soprano pipistrelle relies on fly species, and they also, it's very important that they consume major pests in Ireland and pests that they could have a very economic impact on Irish economy. So it's very good point for the bats. So now I will finish, and so just now it's more future directions that we can discuss that after if you want. So we really need, in my opinion, to assess the economic impact on the foraging behavior on the toucan flies, because I think we can help farmers for that. I think we also need to investigate the diet of other bat species, common bat species like the common pipistrelle or the Leicester bat. And the Leicester's bat is interesting because this species another foraging behavior, so it will be very interesting to look into this diet. And of course, we have urban area in Ireland, and it would be very interesting if we can investigate if they hit mosquitoes or midges in this area, just to see if we can find some interesting prey in the diet, like they could have a benefit for the human health, would be very good too. So yeah, so this is as a project, it's what we found, and thank you very much. So thank you all the volunteers that helped for the 3 years. Without you, it won't be possible. And of course, Tina and Neve and all my team in Butler. But thank you so much for all the volunteers to help for this project.